Now it's really my job to introduce our first speaker, and that is Dr. Trish Simner. Dr. Simner is Associate Professor of Pathology at the Johns Hopkins University School of Medicine and Director of Medical Bacteriology and Infectious Diseases Sequencing Laboratories at the Johns Hopkins Hospital. So welcome on stage, Trish. Good to see you.
Thank you, Justin. Thank you for inviting me here today, and thank you to everybody for sticking around. So today I'm going to be talking about bringing nanopore sequencing into the clinical microbiology setting with targeted approaches. Here are my disclosures. And so my objectives are to discuss not only the advantages of nanopore sequencing, which Justin highlighted so nicely, but also some of the challenges that we face in the clinical setting, and then describe two different scenarios where we've wanted to implement nanopore sequencing for patient care purposes. So as highlighted by Justin, there are many advantages to nanopore sequencing for patient care. The long reads allow for better coverage of a region or a gene of interest like 16S rRNA, better complete whole genome sequencing for genomic analysis. We get better turnaround times due to the real-time sequencing capabilities. There's limited hands-on time due to the simplified library preparation method, so there's ease of use in the clinical microbiology laboratory, and we're starting to see increased automation, which is exciting. And then we also see some benefits in terms of costs, which I'll show you in one of our studies. The challenges are that we do see some higher error rate. However, we're seeing improved accuracy with recent chemistry. We could also overcome some of this with bioinformatics-based methodologies. There's large data storage requirements, so depending on your data retention policies, that's something you need to consider from a clinical laboratory standpoint. And then we see the changing the chemistry. So in the clinical lab, we need to lock down our method. We need to validate that, understand the performance characteristics. So any changes require revalidation. And then a lot of the contracts processes right now are focused most on the research setting than bringing it into the clinical setting. But I will say that the advantages far outweigh those limitations, and we're excited to see nanopore sequencing being implemented for patient care purposes in our laboratory. So the first scenario where we've implemented nanopore sequencing is replacement of 16S RNA gene sequencing by Sanger sequencing with nanopore sequencing for bacterial isolate identification. So Sanger sequencing of the first 500 base pairs of the 16S gene is frequently used to identify bacterial isolates that have either ambiguous biochemical profiles or were unable to identify by MALDI-TOF mass spectrometry, so a proteomic approach. Our 500 base pairs targets the V1 to V3 region. But when diversity does not occur within that region, a longer sequence of the 16S RNA gene or alternative targets like RPOB or SEC-A could be used to distinguish between genus and species. So we were at a point where our Sanger sequencer in the clinical laboratory is towards its end of life, and we're wanting to replace our targeted sequencing on our Sanger sequencer with next-generation sequencing-based methods. So we decided to evaluate the Oxford Nanopore 16S RNA sequencing kit for a replacement for our 16S Sanger sequencing for bacterial identification. And this was a study performed by a previous fellow of ours, Dr. Vicky Campodenico. So we, in the study, evaluated 102 prospective isolates that were sent for Sanger sequencing for bacterial identification, and also challenged it with 51 isolates where we knew the longer read length or coverage of the 16S gene would help resolve the identification. For Sanger sequencing, we used a Boyle Lysis PrepMen Ultra kit, followed by the MicroSeq kit sequenced on the Applied Biosystem 3500, and used SmartGene with the Centroids database for analysis of our results. For nanopore sequencing, we found the Boyle Lysis kit interfered with our sequencing, so we needed to do a manual kit-based method, which becomes important when I talk about hands-on time in a future slide, where we utilized a kit that was already that we already were accustomed to in the clinical lab, the Zymo kit, used the 16S barcoding kit, and in this study we were still using the R10-3 flow cells, which is important with the new chemistry, might see better results than what we were seeing here. We were working closely with SmartGene on this study, where they were helping us develop their 16S app for this nanopore-based data, utilizing the Centroids database for interpretation. We applied the CLSI MM18 guidelines for interpreting the 16S results, as we would for Sanger sequencing for patient care, and then looked at the level of identification that we were getting between Sanger and nanopore sequencing, the concordance of those identifications, the hands-on time, and costs. And so this is just an example report of the SmartGene app. I'm not going to go into too many details here, but this is the app in the cloud-based system. But you also get a report that's generated that will provide the closest identification of your organism, so Rhodococcus here, and then the top 5 matches. It gives you your percent identities and mismatches based off of a consensus building process. What we found was that the average number of reads that we were generating was about 20,000 on average, but it ranged from 68 to about 300,000. We didn't see any good correlation between the number of reads and initial DNA concentration or purity of the DNA, but we did see that it did correlate with certain organism groups. So among the aerobic actinomycetes, mycobacteria, and some of our pigmented gram-negative rods, we were seeing more modest read counts than the rest of the organism groups. In terms of level of identification, as Justin pointed out, we were seeing increased taxonomic resolution, which is not surprising. We're covering the whole gene versus 500 base pairs at both the genus and species level. And when we were getting a similar level ID, the concordance for that ID at the family and genus level was 100% and 87% at the species level. Here's just some examples of where we've seen some increased taxonomic resolution, where we are unable to get an ID by Sanger sequencing, but got a genus level ID by nanopore sequencing, et cetera. So just some examples that we were seeing there. One of our questions, again, turnaround time in patient care, so that we want to get this out as quickly as possible to impact patient care. We wanted to look at how long do we need to sequence the 16S to be able, across these various organism groups, to be able to accurately provide that identification. So at first we looked at what is the maximum read number that we need to generate a stable consensus, and that was 6,000 reads. But that was taking sometimes up to a day to get that sequencing result. So we asked the question, could we take a smaller subset, so let's say 500 reads, how long does it take to generate those 500 reads, and do we see any difference in the identification at 500 versus larger numbers? And we found that we've seen stable ID results at 500 reads, and we could achieve that within 2 hours across the various organism groups that we had challenged the panel with. In terms of workflow and costs, in this study, remember, I really pointed out the fact that we did a manual extraction versus a boil lysis for Sanger sequencing. So the hands-on time were equivalent. We've now automated our extraction method, so those hands-on time were mainly due to the upfront extraction and not actually the sequencing itself. The turnaround time is now same-day results versus by our Sanger sequencing method, we were at 2.5 days before our results were reported into the patient chart. And then we were also seeing cost savings. So if we max out the 24 indices on that run, we're getting $25 for a bacterial 16S identification versus $80 by Sanger sequencing. So in this initial study, we demonstrated that we have similar performance characteristics between Sanger sequencing and nanopore-based sequencing for bacterial identification. Nanopore sequencing provided superior taxonomic resolution at the genus and species level while reducing the costs and turnaround time. So the next evaluation that we had was wanting to see if we could do some targeted sequencing directly from patient specimen. So we were looking at sterile fluids like peritoneal, pleural, and synovial fluids. And we were also wanting to detect fungal etiology. So in addition to 16S RNA, we were looking at the internal transcriber spacer region for fungal identification. And this is in collaboration with our nanopore colleagues. So this is a study that our previous fellow, Dr. David Gass, progressed and had started and had applied for an internal Johns Hopkins grant to start with. So we had implemented metagenomic, untargeted, next-generation sequencing from CSF specimens at Hopkins in 2020, and our plan was to go live with other fluids. But unfortunately, the method that we had optimized and developed for CSF just wasn't meeting the performance characteristics that we wanted from other sterile fluids. And so it's mostly we see a robust host response in these sample types, and it was interfering with our kind of limits of detection or analytical sensitivity. And so we decided that we're either going to need a host depletion method or we're going to try to do a targeted sequencing approach. And most commonly we see bacterial and fungal etiologies, and hence the reason why we decided to proceed with a duplex 16S ITS direct from sterile fluid assay. So this is the workflow that we had generated. So we have a negative extraction control that goes through the entire process. We also have a positive control that goes through the process as well. The negative extraction control is very important because we need to assess what is clinically relevant and interpret those to report to the patient chart. There's background nucleic acid in everything, so you need to understand what is significant versus not. And then you'll see here we did a bead beating for the more difficult to lyse organisms like our yeast and some of our mycobacteria, etc. And then we did some sequencing, multiplexed a whole lot for the study on a MinION R10-4 flow cell, but we also started to play around with the fungal. The fungal is kind of fun because it's cheaper, a quarter of the pores, but it actually, in terms of costs and sterile fluids, we're not going to have large batches, so we're trying to think about smaller batch testing and what could we do with a fungal with the costs of the fungal being a lot more affordable. We get into the range of, some of our multiplex molecular panels when we're thinking about costs per test. So one of the first things that we needed to do was define our positivity threshold here. We looked at reads above alkaligenes, which was the most common contaminating nucleic acid that we see within our nanopore sequencing reads. And then we also looked at either 10 times above the reads above the negative extraction control or 10% above the NEC. And then we also tried to quantify what were the common nucleic acid contaminating reads that we get from our sequencing results. So the first thing, once we kind of started to define that, was trying to understand what are our limits of detection with this assay. And in this particular table, I'm presenting the cutoffs using the reads that are 10 times above the negative extraction control. That was the threshold that performed best in our studies. It's also the threshold that we apply for our Illumina sequencing, so it was nice that we can keep that threshold being consistent. We did spike-ins with negative pleural, peritoneal, and synovial fluids, so we're actually using the matrices that we would use clinically, that would be testing clinically, and we spiked in a gram-negative combo, a gram-positive combo, and a yeast combo, and did from 10 to the 6 to 1 CFU per ml, and did all of this in triplicate, and we defined the LOD as the lowest dilution for which we were able to identify, applying our positivity threshold, the organism that we had spiked in. And so you could see pretty good limits of detection on our gram-positive and gram-negative side. We were seeing quite high limits of detection, 10 to the 4, 10 to the 5 on the yeast side. So we knew right away that yeast detection might be a little bit of an issue as we move forward. The other fun thing that we did is we tested at the limit of detection the same specimens by fungal, and we're able to show very similar limits of detection by fungal as we were by MinION in this study for the 3 different specimen sources. When we got to our accuracy assessment, so this is taking 50 samples of each of our specimen type where that are positive for known organisms or negative, and comparing our 16S ITS duplex results to standard of care. The range in results here really just reflect the different positivity thresholds. We found above the alkaligenes was the best in terms of specificity, but not in, the best in sensitivity, but not in specificity, and the best balance was 10 times above the NEC. It performed best with pleural fluid. It could have just been the samples we had chose here. But again, we were seeing kind of the issues that we were seeing on the LOD studies with candida detections. And so we did see pretty good results with our 16S with the bacterial identification, but we're missing many of our yeast culture positives. This is a study in progress I will highlight. So some of these percent negative agreements or false positives are actually most likely true positives. And that's because a lot of these were previously treated that are patients that were actually able to detect the pathogen that was non-viable by our culture-based methods. And here's just an example of a patient who had a history of IV drug use, presented to an outside hospital with myositis of the pectoral muscle, and represented with necrotizing fasciitis at our hospital. The patient was treated broadly with vancomycin, clindamycin, and piperacillin and tazobactam. Blood cultures and tissue cultures were positive for Streptococcus pyogenes. We had enrolled the pleural fluid into this study as part of our evaluation. The patient developed a pleural effusion, had already been started on antibiotics. They submitted the pleural fluid to the lab. The gram stain was heavy PMNs. No organism seen. The culture was negative, but we were still able to detect the Streptococcus pyogenes by our targeted method, just showing that the pre-treatment impacted detection in the laboratory. And so upon discrepant analysis, a lot of those false positives are likely going to be true positives. So we're still in the process of doing that. I just wanted to highlight that. And then we did see those sensitivity issues. We were not getting down to the limits of detection that we were seeing with some of our culture-based methods, and we found that it really correlated with the amount of host response that we were observing on our gram stain. So we observed higher abundance of hosts among samples with high PMNs on gram stain, which is not surprising. And so the predominance of the hosts reduced our likelihood of detecting the pathogens, even by our targeted approach. So just overall in this study, we've seen similar LODs between MinION and fungal-based testing, which was fun to play around with the fungal a little bit and see the capabilities. The 16S ITS duplex demonstrates a potential as a diagnostic tool. It doesn't have the sensitivity as some of our culture-based methods, but it can be used as an adjunct. And this is often the way we're using these tests right now in the clinical lab, is when things are unrevealing by our standard of care methods, we proceed to next generation sequencing based modalities, where this could be helpful in cases where potentially the patient was previously treated and we want to identify the potential etiology. And we also are going to evaluate future approaches to reduce the amount of hosts being sequenced to help enhance our sensitivity. So again, shows the promise of this, but still a little bit of work to be done. So overall, replacement of the 500 base pair 16S rRNA Sanger sequencing with the 1.5 kb ONT kit for bacterial identification provides higher taxonomic resolution. Targeted 16S with rRNA and ITS duplex from sterile fluids is a promising approach, but requires a little bit additional troubleshooting to achieve those lower limits of detection. But overall, nanopore sequencing provides a rapid, easy, and affordable solution to targeted sequencing in the clinical microbiology laboratory for patient care. So I want to thank everyone at Hopkins, SmartGene for the analysis on the bacterial side, Aris Genetics for the analysis on the 16S ITS duplex side, and of course our colleagues at Nanopore who help support some of this work as well. So thank you so much for your attention.